Hello everybody, today's topic is hybridization. Today we will discuss about what is hybridization, how it is useful in plant breeding. Okay, first we come to history of hybridization. Rudolf Jacob Camerus was the first who reported reproductive part as male and female in plants. First artificial hybrid was produced by Thomas Fairchild. He crossed Dianthus barbatus to Dianthus caryophyllus, and the hybrid Dianthus caryophyllus barbatus was also known as Fairchild Mule. Joseph Gottlieb Karlreuter has produced hybrid tobacco plant between 1760 to 1766. Hybrid variety of apple, grapes, peaches and blackcurrant was produced by T. A. Knight between 1759 to 1835. Sargret, Charles Nodin, John Gross, Carl Friedrich von Gardner noted uniformity in first generation variety F1, dominance in F1 and separation and appearance of parental type in F2. Later on, Geiger John Mendel proposed a law of inheritance in 1865, and these all discovery contributed to the certification of hybridization processes. Now we should know what is hybridization. Its actual definition. Hybridization is the process of crossing two genetically different species to create a heterozygous F1 variety. Hybridization occurs when inbreed parents are mated or cross-pollinated. It creates a heterozygous individual. Its benefit is to increase heterosis, that is hybrid vigor in F1 generation. In this diagram, you can see that inbreed A and inbreed B both are a small heighted plant, and the hybrid which is in F1 generation is a tall plant. This has increased the vigor or the quality of plant. So it is the benefit of hybridization that it produces heterozygous individual which can be better than both of its parent. Objective of hybridization. The main objective of hybridization is to produce genetic variation. In previous lectures you have seen that for selection and for introduction and for domestication, genetic variation is the must, and so hybridization is a manual process to produce genetic variation. So the main objective of hybridization is to produce genetic variation. The degree of variation depends on the number of different type of gene present in the F1 parent. Okay. The generation obtained from crossing of F1 plant are often termed as segregating generation. Hybridization aims to create a new reservoir of genetic variations to enable for the selection. The major objectives of hybridization are achieved by number 1, combination breeding, number 2, transgressive breeding, and number 3 is production of hybrid. We will study these all in detail. Now come to combination breeding. Combination breeding is the transfer of one or more character from other varieties to a particular variety. These character may be oligogenic or polygenic. In this way, genes for disease resistance, quality, traits, etc. can be transferred. This transfer enhances desirable character of offspring, such as tiller number, grain per panicle, seed weight, etc. Combination breeding are applied as in backcross method, in pedigree method. We apply this combination breeding. We will study these methods in detail in further classes. Now come to transgressive breeding. Transgressive breeding is based on transgressive variation of character in segregating generation like F2. And now you should know what is segregating generation. You know, when we cross P1 and P2. P1 and P2 are parents. Okay. When we cross these parent, then the progeny which we get in first generation, it is F1 generation. So it is the character in this will be expressed. It become heterozygous, and the character it expresses maybe if it express dominant character. Okay. Uh, then when it is further inbreeded, then what we get in next generation is the F2 character. And you know, in first generation, if we get all the dominant and we cross it by Mendelian law, you know that next is segregating generation, means the character are distributed. Like if we have crossed in P tall and short plant, the F1 in F1 generation all the plant is tall, but in F2 generation one will be tall, one will be short. Okay. And 1:2, 2:1 ratio will be genetic ratio, or although it will be phenotypically it will be 1:3 ratio. Okay. So if from genetic level we can segregate these characters. So transgressive breeding is based on these segregating character. So it starts with F2 generation. A cross is made between two strains of plant, and the F2 is screened for transgressive variation. So in this, F2 will be the main plant to further study or further do further study. Transgressive segregation is segregation of character beyond the parent limits in the segregating generation like F2. Pedigree method and population approach is designed to carry transgressive breeding. Third objective of hybridization is to produce hybrid. You know that if P1 and P2 parents are cross, we get F1 generation. F1 is hybrid, and the crossing is made to get a better variety, and it is, we call it heterosis and hybrid vigor. F1 seeds can be raised in bulk through hybridization and distributed directly for cultivation, especially in cross-pollinated crops. Now come to types of hybridization. On the basis of cell type involved in crossing over, hybridization can be divided into two types. Number one is hybridization in sex cell. Number two is somatic cell hybridization. On the basis of taxonomic relationship of the two parent, hybridization may be divided into two types. Number one is intervarietal hybridization. Number two is distant hybridization. Intervarietal hybridization is further divided into intraspecific hybridization and interspecific hybridization. And distant hybridization is further divided into intrageneric hybridization and intergeneric hybridization. Intraspecific hybridization. In this, crosses are made between two individuals of same species. This type of crossing is done to improve self-pollinated crop. Interspecific hybridization. When crosses are made between individuals of two different varieties of the same species, then it is called interspecific hybridization. This type of hybridization are made to improve self-pollinated as well as some cross-pollinated crops. Distinct hybridization. In this, parents involved belonging to different species of the same genus or of different genera. We have already studied that it is also of two types, intrageneric and intergeneric. Intrageneric hybridization. When crosses are made between two individuals of different species belonging to the same genera, then it is called intrageneric hybridization. It is used to produce resistant varieties from disease, pest, or frost or drought, etc. Intergeneric hybridization. When crosses are made between two individuals of different genera belonging to the same family, it is called intergeneric hybridization. It produces resistant varieties as well as desirable combination of all the character. For example, we have Raffino brassica. It is crossed between brassica and Raffinus belonging to same family Brassicaceae, but these both are of different genera. Another example is Triticale, which is a hybrid between wheat and rye. Here it is an example of interspecific cross. It is a sterile plant of Trillium hybrid. It is crossed between Trillium cerium and Trillium grandifolium. In this photograph, you can see somatic hybridization. When the somatic cells of two different species or variety are mixed in their protoplasm and then a new variety is created, it is called somatic hybridization. Somatic hybridization can be defined as development of hybrid plants through the fusion of somatic protoplast of two different plant species or varieties. In this slide, somatic hybridization techniques has been described. Although it is a tough technique. Number one, first step is isolation of protoplast. Second step is fusion of protoplast of desired species or variety. Number third is identification and selection of somatic hybrid cell. Number four is culture of the hybrid cells. And number five is regeneration of hybrid cell. Now we should see all these in details. First step is isolation of protoplast. Isolation of protoplast can be done by two methods, or isolation of protoplast is separation of protoplast from the plant tissue. It can be done by mechanical method or by enzymatic method. Although mechanical method is quite tough and it is inefficient in comparison to enzymatic method. In this slide, process of mechanical method has been shown. First we take plant tissue, then we do cell plasmolysis, and then we observe the cell in microscope. Then the cell wall is cut with knife. It is a microscopic and small knife, and then it is release of then the protoplast are released of both the plant. Then this is there is collection of protoplast. Merit of mechanical method is that it can be used for vacuolated cells like onion, bulboscale, radish, etc. But it has demerits also because it is low yielding of protoplasm. It is very laborious and tedious process. The protoplast viability is also low in this method. Now come to techniques of enzymatic method. First, the leaf are sterilized and the epidermis is removed. The plasmolysed cell are then collected and it is treated with pectinase and cellulase enzyme in combination, and then protoplasm are released, and this as released protoplasm are isolated. and it is done by another method in which plasmolysed cells are treated first with pectinase, then there is release of isolated cells, and then the isolated cell are treated with cellulase enzyme, and then protoplasm is released, and then it is further isolated protoplasts are collected. Merits of enzymatic method. It is used for a variety of tissues and organs such as leaves, petioles, fruits, roots, stem, shoot apex, embryo, microspores, etc. It is high yielding of protoplast. It is very easy to perform in comparison to mechanical method. In this, the protoplast viability is more. Next step is in somatic hybridization is protoplast fusion. Fusion of protoplast of two different genomes is called protoplast fusion. It may be spontaneous fusion and it may be induced fusion. We will discuss all these in detail. In spontaneous fusion, it may be intraspecific or it may be intergeneric. And induced fusion, it may be of three types: chemofusion, mechanical fusion, and electrofusion. Spontaneous fusion. When protoplasts are fused spontaneously during the isolation process, it is called spontaneous fusion. It is mainly due to physical contact. It is of two types: intraspecific and intergeneric. Intraspecific produces homokaryons, and it is viable, while intergeneric has no importance as it does not survive. Now come to induced fusion. It is of three types: chemofusion, electrofusion, mechanical fusion. Chemofusion is fusion of protoplast which is induced by some chemicals. It may be types of fusogen, it may be polyethylene glycol, it may be sodium nitrate, it may be calcium ion, or it may be polyvinyl alcohol, etc. Mechanical fusion. Physical fusion of protoplast under microscope by using micromanometer and perfusion pipettes is called mechanical fusion. And next is electrofusion. When fusion of protoplast is induced by electrical stimulation, then it is called electrofusion. Then next step is identification and selection of some somatic hybrid cell. The hybrid are identified on the basis of difference between the parental cell and hybrid cells. There are several markers for selection of hybrid. These may be by genetic complementation, by phytotoxins, specific amino acid, auxin, autotropy, antibiotics, auxotropic and metabolic mutant, chemosomal analysin, and herbicides. The next step is culture of hybrid cells. Hybrid cells are cultured on suitable medium and provided with the appropriate culture condition if matters. When the hybrid is selected, then the next step is its regeneration. So the next step is regeneration of hybrid plant. The plant are induced to regenerate from hybrid cell. These hybrid plant must be at least partially fertile. In addition to have some useful property to be of any use in breeding scheme. Advantages of somatic hybridization. Number one is production of novel interspecific and intergenic hybrid. For example, pomato, which is hybrid of potato and tomato. Next is production of fertile diploid and polyploids from sexually sterile haploid or triploid or aneuploid. Next importance is transfer gene for disease resistance, abiotic stress resistance, herbicide resistance, and many other quality characters. It is utilized in production of heterozygous lines in the single species which cannot be propagated by vegetative means. It is used in study for the fate of plasma gene. It is utilized in production of unique hybrid of nucleus and cytoplasm, which was not possible otherwise. There are some limitations of somatic hybridization. Number one is poor regeneration of hybrid plants. Number two is it is non-viability of fused product. It is not successful in all plants. The production of unfavorable hybrid are also possible. Lack of efficient method for selection of hybrid is one of the limitations, and there is no confirmation of expression of a particular trait in somatic hybrid. That is also its limitations. In the previous slides, you have seen the process of hybridization. You have seen that it is quite time-consuming and tedious process. Even though we like hybrid plants and hybridization is done in selected plants. You can see here tomatoes plant, hybrid tomatoes. You have seen hybrid hibiscus, and you in this we have taken the process. So although the technique is very tough, even though as the result is very fruitful, and we need it, so we are interested in hybridization. It is also useful in cross-pollinated plants, but it can also be used in self-pollinated crops also. Hybridization method of plant breeding and self-pollinated groups. There are several methods of improvement of self-fertilized plant by hybridization. These are pedigree method, bulk method, single seed descent method, and backcross method. These all methods are based on hybridization technique, and all the cross-pollinated method are based on hybridization technique only. That is all for today. Thank you.